Welcome back to JLM Bio. Today's module is the renal system. In this session, we'll cover the following learning objectives. Identify the structures that compose the urinary system and describe the general function of each. Explain the relationship between collecting tubules and two types of nephrons and collecting ducts. Define glomerular filtration rate and the factors that influence it. Identify the location and describe the structure of the JG apparatus, the juxtaglomerular apparatus. Differentiate among filtrate, tubular fluid, urine, and list the urinary system structures that transport them. Define net filtration pressure and which pressures constitute it. Compare and contrast the myogenic response and the tubular glomerular feedback mechanism. Explain what is meant by the countercurrent multiplier that occurs within the nephron loop. Explain the contribution of urea cycling to the concentration gradient. Describe how the resorption of sodium, potassium, calcium, and phosphate occurs. Explain ball volume regulation by the renal system. And explain the renin-angiotensin-aldosterone pathway. Anatomy of the urinary system. The kidneys are two symmetrical bean-shaped reddish-brown organs, each containing a hilum, that is a biconcave border, where there are vessels, nerves, and the ureter connecting to the kidney. The left kidney is positioned between T12 and L3, and the right kidney is about 2 centimeters inferior to it. The features include the dense, irregular connective tissue layer that anchors the kidney to the surrounding structures, also known as the renal fascia. There's also an adipose connective tissue layer that provides cushioning to the kidneys, called the perinephric fat layer. And on the outside, we also have this capsule that directly covers the outer surface of the kidney. And then underneath that layer, we have the renal capsule, which directly covers the outer surface of the kidney. So it goes from renal fascia, perinephric fat capsule, and then the renal capsule. The sectional anatomy of the kidney. The outer region of the kidney's parenchyma is also known as the renal cortex. And then the inner region of the kidney's parenchyma is called the renal medulla. There are extensions of the cortex that project into the medulla called the renal columns, and the subdivisions of these columns are called the renal pyramids. The portion of the kidney where the external edge of the medulla meets the cortex is also referred to as a junction. Each kidney has a medially located space called the renal sinus, in addition to the parenchyma that serves as a urine drainage area. Each of these renal sinuses are then organized into minor calyces and major calyces, and eventually the renal pelvis. Each of the 8 to 15 funnel-shaped minor calyces is associated with that renal pyramid. There are several minor calyces that merge to form 2 to 3 major calyces, and then the major calyces form the funnel-shaped in towards the renal pelvis, where it drains into the ureter. The ureters are these long fibromuscular tubes that transport urine. They originate from the renal pelvis of the kidneys, and they enter into that posterior lateral wall of the bladder called the ureters. They enter in the posterior lateral wall of the bladder. The innermost transitional epithelium of the ureters is both distensible, stretchable, and impermeable to the passage of urine is the mucosa. The middle layer of this ureter is composed of mostly the longitudinal and outer circular layer of smooth muscle that helps enable peristalsis, the movement of urine. And then the external layer is composed of mostly collagen elastic fibers called the eventia. This keeps it intact and supports the flow of urine in one direction. The urinary bladder. The urinary bladder is an expandable muscular retroperitoneal organ that serves as a reservoir that holds our urine. The posterior inferior triangular area of the urinary bladder remains immobile, and as the bladder fills up, it's going to evacuate and functions as a funnel for urine into the urethra. This region is called the trigone. The innermost mucosal layer is transitional epithelium that forms these folds called the ruga, and the middle layer contains a muscle called the detrusor, which can drive the output of urine. That thick muscle near the end of the urethra is called the internal urethral sphincter. And be aware that the epithelium does transition into stratified squamous near the urethra's opening for protection. Eventually, the urine is going to make its way out through a ring of skeletal muscle on its way to the toilet via the external urethral sphincter. The female urethra. The urethra is an epithelial-lined fibromuscular tube that exits the urinary bladder. It transports the urine to the exterior of the body. Its involuntary superior sphincter of the urethra is going to be called the internal urethral sphincter. The female urethra is lined by mostly stratified squamous epithelium and is about 4 centimeters or so in length, functions specifically just to transport urine from the urinary bladder to the outside world. On the other side, the male urethra is a lot longer. It's about 20 centimeters. It's the most dilatable portion of the urethra, and it's lined by transitional epithelium, and this region is called the prostatic urethra. And the function here is that it transports both urine and semen. The blood flow of the kidney. At least 20 to 25% of the resting cardiac output normally flows through the kidney for the purpose of removing unwanted waste. The renal arteries arise from the abdominal aorta, and they function to deliver blood to the kidney. The arteries branch from the renal artery are called the segmental arteries. These arteries then branch from the segmental arteries and travel through their renal columns toward the cortical medulla junction, are then called the interlobar arteries. These arteries then branch from the interlobar arteries and project parallel to the base of the medulla, and they become the smaller arcuate arteries. Then finally, these arcuate arteries extend into the interlobular branches and project into the cortex. Eventually, you're going to see the microscopic structure of the afferent arterial branch from the interlobular arteries into the glomerulus. Each of these arterioles enter the renal corpuscle, forming the glomerulus, where the blood is then filtered and eventually exits the renal corpuscle through the efferent arteriole. So the afferent arteriole is the entrance in to the renal corpuscle, and the efferent arteriole is the exit of the renal corpuscle. The functional anatomy of the kidneys encompasses structures that include nephrons, collecting tubules, collecting ducts, and their associated structures. The microscopic filtration unit of a kidney is called a nephron. Each nephron consists of two major structures, a renal corpuscle and the renal tubule. The renal corpuscle, and the most of the renal tubule, except for the nephron loop, resides inside the cortex of the kidney. All of these regions are inside the cortex, whereas the nephron loop is inside the medulla. The bulbous region of the nephron located within the cortex is called the renal corpuscle. As you can see right here, this is the renal corpuscle, which is composed of the glomerulus and the glomerular capsule. The glomerulus is a conglomeration of capillary loops called the glomerular capillary. Blood enters in the afferent arteriole and exits out the efferent arteriole. The glomerular capsule is also referred to as the Bowman's capsule. It's the same thing. The glomerular capsule is formed by this internal impermeable layer directly overlining the glomerular capillaries, and is mostly composed of simple squamous epithelium. There's also a tiny amount of space between the visceral and the parietal layers, which receives filtrate. This is called the capsular space. Then we have the renal tubules, which make up the rest of the nephron. This is going to be most simple epithelium, and it consists of three continuous sections. The first part of the renal tubule is called the PCT, proximal convoluted tubule. This region is composed of mostly simple cuboidal epithelium, tons of microvilli that increase the surface area, and it helps with reabsorption. Then the next part of the renal tubule is going to be the loop of Henle, right here. The loop of Henle, also known as the nephron loop, is composed of a descending limb and an ascending limb. The descending limb extends into the PCT to the tip of the nephron loop, while the ascending limb returns back to the renal cortex, terminating at distal convoluted tubule right here. It should be noted that the descending thin limb right here of the loop of Henle is composed of mostly simple squamous epithelium, whereas the thick limb right here, the ascending limb, is composed of mostly cuboidal. So the simple squamous, and this one is going to be simple cuboidal. Then the region of the renal tubule originating from the thick ascending limb of the nephron loop is lined with small simple cuboidal epithelium, is then terminated when it becomes the DCT, the distal convoluted tubule, where there's also going to be some microvilli as well as ability to reabsorb some fluids. So in summary, the proximal convoluted tubule, PCT, is located entirely in the cortex. It's outlined by simple cuboidal epithelium with microvilli. The descending tubule, DT, descends into the medulla, alternates between the thick and thin segments, connects the ascending and descending tubules called the loop of Henle, and then the ascending limb alternates between the thick and thin segment, which is mostly composed of cuboidal epithelium. And then the final part is going to be the distal convoluted tubule, DCT, which again is going to be simple cuboidal epithelium. However, it does not have microvilli. So collecting tubules and collecting ducts. Several nephrons drain into collecting tubules, and a series of collecting tubules drain into the collecting ducts. A group of collecting ducts then drain eventually into the papillary ducts within the renal papilla. Epithelial cells of the collecting tubules are mostly cuboidal shaped, while the collecting ducts near the renal papilla are very tall and columnar. The collecting tubules and collecting ducts contain two types of specialized cells. The first type is called the principal cells. The principal cells have receptors that can bind hormones called aldosterone and ADH, anti-diuretic hormone. Another type of cell is called the intercalated cells. The intercalated cells have two subtypes. We have type A and type B. Though both of these help regulate urine and blood pH. Type A's role is to remove protons, whereas type B cell function is to remove bicarbonate. The juxtaloid glomerular apparatus. The region of the nephron that regulates the filtrate formation and systemic blood pressure is called the juxtaloid glomerular apparatus, also known as JGA. The DCT, in contrast with the afferent arteriole of the same nephron, creates a specialized region. The smooth muscles of the afferent arteriole that contract when stimulated by stretch or the sympathetic division can synthesize, store, and release a compound known as renin. The cells that can release renin are called the juxtaglomerular cells. Then we also have a group of epithelial cells inside the walls of the DCT that are come in contact with these juxtaglomerular granular cells of the afferent arteriole. These are called the macula densa. The macula densa cells can detect changes in sodium concentration within the lumen of the DCT and signal the granular cell release renin. So these guys monitor the amount of cell concentration, and they can stimulate the juxtaglomerular cells to then release renin. Renin is then used to activate a pathway to eventually cause the release of aldosterone. This will then activate, and then this will activate aldosterone, and which will help increase water uptake and increase sodium uptake.